Good afternoon. Good afternoon. How's everybody? Happy Monday. All right, while we're waiting for everybody else to come in, you can take a look at the new unit, genetics. This one is going to be a heavy hitter. This one we're going to need you to pay close attention. There's a lot of activities with this. Lots and lots and lots of vocabulary. So please take a look at that while we're waiting for everybody to come in. Also, I've assigned, and I really just want to, let me just wait till everybody comes in. So again, you should go to Canvas and it's published. So take a look at the next unit. We're finished with cells and mitosis. We're still going to be talking about chromosomes. So it's going to be real important that we talk about chromosomes in this unit because genetics is all about what the chromosomes do and how they do it and how they are expressed. So now we're going to take the chromosome under the microscope and talk about genes. Folks are very late today. All right, well, they're late, so I want you to go ahead and get started. Click on, go to your resources and click on progress learning. You should have an assignment for progress learning. Please let me know if you, all right, progress learning, progress learning. So I told you that what we are going to do is start spending a few minutes at the beginning of class doing practice questions. And so you've been assigned some questions. Mm, 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 mm. Okay, that's not what I want. I want the class assessment. Okay, now folks are coming. Y'all are late. All right. So in progress learning, you should see that you've been assigned a bell ringer on mitosis and meiosis. So please log into that. Everybody needs to log into Progress Learning. If you're not able to log into it, please let me know, but you've been assigned a bell ringer question or a set of bell ringer questions. So, and again, these are practice. We're going to be practicing questions. So everybody log into, I'm gonna stop sharing for a minute, log into Progress Learning. All right, so I'm and I'm looking at GoGuardian, and two people have logged into Progress Learning. The rest of y'all just sitting there. Again, if you bother logging into class, please do classwork. All right, go to Clever, log into Progress Learning, and you've been assigned a bell ringer set of questions. Please work on those right now. Progress Learning, Progress Learning. Again, I got 5 people who are logged into Progress Learning. If you are logged in right now, you should be in Progress Learning. Progress Learning. All right, so you have to do just for the next 10 minutes. I'll let you work on that, and then in 10 minutes, we'll start genetics. So when you finish that, come back to Canvas and look at the unit on genetics. 10 minutes, 10 minutes. So that's at 12:40. Just about to mark these folks late. All right, good afternoon. If you just came in, we are in Progress Learning. You've been assigned an assignment in progress, bell ringer question in Progress Learning. That's what everybody needs to be doing for the next 8 minutes now. All right, again, for the next 7 minutes, everybody's in progress learning. So far, I only have one person who's completed. That's Chris. Everybody else, I don't see any action going on. Everybody needs to be in progress learning. You got 2 more minutes because we're going to move on. Okay, folks. It's not a test, but it's practice. Remember what I told you we are going to do? Most of you did not score very well on the benchmark. So these are practice questions. So yes, they're mandatory, but they're not a test. They're practice questions. And we're going to be practice questioning every class. So there will always be a bell ringer question for you to practice on. All right, so I picked the one that we just completed because again, on the major tests, on the benchmark test, those were questions that you at the time probably couldn't answer properly because we had not covered it as a unit. But now that we have covered it as a unit, those are the questions that should be easier for you to answer. What was I going to say now? Thought left my mind. All right, so I picked meiosis and mitosis because we had just completed them. So being able to answer the question about budding and regeneration, you should know how to do that. All right. So we're going to move on now, folks. So you can come back to, if you haven't finished the progress learning, we're going to come back to it. Or actually, you're going to need to finish that on your own time because we'll be keeping score and I'm going to go back and I'll call you into afternoon tutoring if you don't get it done. So we need to get this done when it's allotted in class time. All right. So we're starting genetics. So again, we are looking at chromosomes and when we were doing protein synthesis, I told you to think about a chromosome as a string of pearls with little beads on it. Okay, and each bead represents a different gene or is coding for a different characteristic. So different characteristics exist on different chromosomes. So this is why we were talking in the last unit about sister chromatids and sister chromosomes, because certain sets of chromosomes have certain pieces of information. So while humans have 46 chromosomes, not every one of the 46 chromosomes has the same information on it, which is why if you have damage to one, it creates one type of mutation and it's not entirely bad for the body. Some of them can be lethal because they can lead to diseases that eventually will be detrimental to your health. But um, we're going to look at uh, sorry, I just let somebody in. We're going to look at what these genes do and what does it mean to inherit. And we're going to be looking. So again, you're going to need to pay attention because this one is we're going to learn how to do Punnett squares, which look at predicting heredity. So you're going to have to learn how to do that. You're going to have to learn how to be able to predict traits. So I need you to pay attention. I need you to not be playing games or logged in just for logging in sake, because this one is a little more difficult and you got to keep up. You got to keep up. And there's a lot of vocabulary with this.
Created using Powtoon.
All right. All right. One of the people that you have to know, usually with certain things with biology, there's always the father of something. That means the person who is responsible for doing all of the studies on this subject that now we now follow their plan. Okay. So Gregor Mendel is that person for genetics. So when you hear the name Mendel, you need to know that we're talking genetics. And what he did is he looked at, there are what we call probabilities of certain characteristics showing up in a person. And there's two ways that a thing can show up. I mean, the genes or the characteristics are called traits, and traits can show up physically. That means something that we can see. They can show up behaviorally, which is also something we can see, but also what we call genetically. They show up. So we're going to talk about phenotypes and genotypes. Those are going to be two words we're going to use a lot, a lot, a lot. Genotypes and phenotypes. What we can see physically and what the person has genetically that you can't see. Okay. All right. So again, traits are located on your DNA. And again, think of the DNA as a long chromosome that's each one is carrying a trait for particular characteristics such as height, hair color, uh, skin color, eye color, all of those types of things. Um, because of what they know about genetics now, they can do what they call selective breeding. They don't necessarily, and actually there is such a thing now as called designer genes. So people do who do artificial insemination, they can look at a gene profile and decide that these are I want somebody that has the following genes. And so they are designing what they want their offspring to look like. Those are for the people who are doing IV fertilization. So you look at what you have and you look at what you would like, the characteristics that you want your offspring to have, and then you select someone who has those characteristics, and then you can selectively breed the individual. So most of the time when we think of selective breeding, we think of plants and dogs and horses and things of that nature. Like the horses that they breed for the Kentucky Derby, they breed them so that they come from parents that are good runners and are more likely to win the race. With plants, they want to create crop that are strong crops, resistant to bugs and all that sort of thing. So they are selecting the characteristics um, that they want that individual or that plant to have. And in order to do that, you need to know a little bit about the traits that they have. We need to know which ones are dominant. Those another word we're going to learn, dominant, and which ones are recessive, which ones are going to show up physically, and which ones are just going to be carried genetically. All right, so I'm going to go ahead and start this.
Gregor Mendel is known as the father of modern genetics. Mendel was an Austrian monk, and he lived from 1822 to 1884. His genius was that he noticed differences in peas being grown in the monastery's garden and applied the scientific method to determine the cause of these differences. Mendel chose the garden pea Pisum sativum because peas were easy to grow, they had a short generation time, they had a number of contrasting traits that were easy to distinguish, such as short versus tall, or white versus purple flowers. And they also normally self-pollinated but could be cross-pollinated. Mendel was able to control mating this way by removing the male or the female flower parts and cross-pollinating with a paintbrush. Normally peas do cross-fertilize or fertilize themselves, but he didn't want this to happen. Mendel didn't really know about inheritance. No one had great ideas about it at that point in time. We knew what cells were, but we did not know about organelles, protein synthesis, or even genes or chromosomes yet. All Mendel had to go by was physical traits and his knowledge of flowers and statistics.
All right. When they say cross-pollinating, in the last class we talked about the A flowering plant has the male parts in the pollen and the female parts in the ovaries, and then the pollen can just fall off um, of the pollen stem and fall into the ovary part, and the plant becomes fertilized. So that's what they call self-pollination. Okay, the pollen falls on the same plant, pollen falls off the male structure, falls into the female structure, and it is fertilized. So what he did was remove those so that they could not self-pollinate, that he would have to pollinate them physically, so that he would have to select the kind of pollen with the kind of characteristics he wanted to pollinate the flower. So that's what they mean by normal pollination and self-pollination. So cross-pollination means I'm going to take the pollen from this plant over here, which is different from this one over here, and I'm going to fertilize it with a different pollen grain, not the one that came from the plant itself, but a different flower. So that's cross-pollination. Just taking and then again, the sperm cells in a plant are in the pollen. The egg cells in a flower are in the seed or the ovule. Okay. All righty. They went through. Okay, before you could do that, they just jumped right into recessive. And so hold on one second. I do have a worksheet for you, but they haven't talked about the words that they have on your vocabulary list. All right, so again, vocabulary, vocabulary, vocabulary. It's a lot of vocabulary with this unit. So before we go on, we have to look at what he discovered, that there are genes that are expressed and there are genes that are not expressed, but they are there. That doesn't mean that they're not there. They're just not expressed.
When I was a little kid, I had this art teacher. She had Miss Frizzle energy and a room full of art. And one of her favorite things to say was, "You've got to express yourself. Art is a way you express yourself in the art." I had her all through elementary. She started saying this when I was like 7, and the phrase didn't have much meaning for me at first. She told us music was another way, and dance. I wasn't really into those either. It wasn't until a lot later I got into poetry, which I love. That was my creative expression. My mitosis poem will catch on someday. Anyway, the word express stuck with me. As a kid, I understood it was a way to take the creativity we all have inside of us and let it out. But that's creative expression. Our cells have expression too, gene expression. They can express genes. To express a gene means that a gene can be used to make something functional, often a protein. In our protein synthesis video, we talk about these steps. The gene, which is made up of DNA, can be transcribed into mRNA during transcription, and then used in translation to make a polypeptide chain. A protein is made up of one or more of those genes.
All of this part is supposed to be review. So up to this part, you need to try to remember what you learned like in December. Okay. But we're still going to need to understand it in order to do this unit.
But you know what? It's not doing that with every single gene. Not every gene is expressed. That's why the phrase gene regulation gets put with gene expression. because the expression has to be regulated. A cell in the eye has no need for using a gene that codes for stomach acid, even though the gene is present. It would be wasteful to express that gene and likely uncomfortable. Prokaryotic cells and eukaryotic cells both have genes to express. Some differences, though. Recall that prokaryotic cells just have the DNA in their cytoplasm. They don't have a nucleus. So transcription and translation all just happen here. Unlike in a eukaryotic cell, there's no traveling of the DNA out of a nucleus. Nope, in prokaryotes it's all right there together and can really both be happening together at the same time as there's no nucleus involved. For prokaryotes, a lot of the control of whether a gene is going to get expressed or not tends to involve or impact transcription. Eukaryotes also have gene regulation that involves transcription, although they'll have a lot of other times for gene regulation too that we'll get into soon. So you might wonder, how does gene regulation involve or impact transcription? Transcription, again, is when an enzyme called RNA polymerase makes mRNA from a DNA template. There are regulatory proteins that can decrease or increase transcription, and that's important among both prokaryotes and eukaryotes. These regulatory proteins are often referred to as transcription factors. Some transcription factors bind to a DNA region called the promoter to help RNA polymerase start transcription, while other transcription factors can bind there to repress it. Some types of transcription factors can bind to enhancer sequences, where they increase transcription, and enhancer sequences can even be far away from the gene. Some transcription factors or other proteins can bend the DNA to bring the promoter and enhancer closer. Ultimately, transcription factors play huge roles in whether a gene is expressed, and we also want to mention environmental factors can influence transcription factors, meaning that the presence or absence of an environmental factor could impact gene expression. Prokaryotes commonly have operons, which feature a regulated cluster of genes along with a promoter and an operator, and they make great examples of gene regulation during transcription. We have an example of this in our Order of the Operon video. But here's a quick recap of just one type of operon known as a lac operon. Here's the repressor. The repressor blocks RNA polymerase from doing transcription, and it does this by binding to a sequence called the operator. Since the RNA polymerase can't work, ultimately these genes are not going to get expressed. That's because the genes are not going to be transcribed into mRNA, so it will not go to translation for a protein to be built. And that's great when the protein isn't needed. This is how the bacterium is regulating the genes in this operon. But sometimes the bacterium may need these genes expressed, and that's because these genes code for specific proteins, including an enzyme that breaks down a sugar called lactose. So when lactose is around, the bacterium wants that enzyme because it wants to break the sugar lactose down. It can eat the broken down lactose. So how does the bacterium turn a gene on? How does the gene get expressed? Well, as you can imagine, this repressor has got to be moved. It's in the way of transcription happening. And it turns out a form of lactose, technically an isomer of it, binds to the repressor, which makes it get out of the way. When the repressor is not blocking this RNA polymerase, transcription can happen. The RNA polymerase does transcription, and it makes the mRNA, and translation can follow, and it uses that mRNA to make protein, which includes an enzyme that can break down lactose, and other proteins too involved in using lactose. So these genes are being expressed when lactose is around. Expression is not happening when lactose is not around. It's a gene regulation example, controlling whether genes are expressed or not. And still on the subject of gene regulation that can impact transcription, we want to mention from our epigenetics video that there can be epigenetic marks on DNA that can ultimately impact transcription. Eukaryotic DNA is often packed by winding the DNA around proteins called histones. There are chemical groups, like methyl for example, that can influence how tightly that DNA is packed. If the DNA is really tightly packed, transcription factors can't really bind in there very well. So if these factors can't bind, then transcription doesn't happen, as shown here with this highly methylated area. But in this case, if methyl is removed, then that would allow the DNA to be transcribed and eventually be used to make protein. So it could then be expressed. And just a note, this can also occur in prokaryotes, although there are differences, for example, in the DNA packing. Now, remember, we said that prokaryotic cells tend to have gene regulation focused around transcription. Eukaryotic cells, however, tend to have more potential opportunities for gene regulation. For example, it's also common for eukaryotes to have gene regulation that is post-transcription, but before translation, during translation, and even post-translation. We'll go through a few examples. Let's have an example of eukaryotic gene regulation that is post-transcription, but before translation. So let's say an mRNA was made in transcription. Eukaryotic cells tend to do significant processing of this mRNA afterwards, and certain areas of the RNA are actually removed. Introns are RNA portions that are removed. This is a type of regulation because if they're cut out, then they're not going to be expressed. But also, it can impact the portions that remain. The portions that remain are called exons, and those portions will go on to translation. Let's have an example of eukaryotic gene regulation that impacts translation. So remember, for translation, you have the mRNA, you got the ribosome, and you have the tRNAs. So there's a little protein called EIF2, or its fancier name, eukaryotic initiation factor 2. Its job? Well, it helps translation get started, hence the initiation. Important for ultimately making a protein, but EIF2 itself can be regulated. If EIF2 is phosphorylated, which means it has a phosphate group added to it, that makes its shape change, so it can't do any initiating. No initiation means no translation, so no protein made. The gene is not expressed. Finally, there's even post-translation regulation, meaning it comes after translation. Chemical groups can be added to proteins or removed from them, which can change where they end up located or how they function, therefore impacting their expression. Like we mentioned for transcription, environmental factors like limited nutrients or UV exposure can influence those modifications. And then there's also ubiquitin. Attach that to a protein, and that can signal the protein to be degraded. So that's going to affect expression too. Let's recap all we've talked about. We mentioned what gene expression is. It's when a gene is used to make a product, which is often a protein. We talked about how gene regulation is a related term, as many times whether a gene is expressed or not is often something that can be controlled or regulated. Prokaryotic cells tend to have gene regulation mechanisms that impact transcription. We talked a bit about an operon, as it makes a great example. Eukaryotic cells can have gene regulation during or impacting many potential points, but also post-transcription, translation, post-translation, and we gave a few examples. So why does this matter? Well, not only does it really matter for understanding how genes in our bodies are expressed, but gene expression is also important to understand when cells in the body are not functioning correctly. Cancer cells, which we've talked about before, can have genes expressed that should not be, or not have certain genes expressed that should be. One example is a cancer cell with a mutation that increases transcription factor activity. If this boosts the transcription of genes that speed up cell division, it could contribute to cancer cells dividing over and over. Gene regulation and gene expression are very important to developing treatments for many conditions. Well,
right, a lot of that was review. That wasn't exactly the one that I wanted you to watch, but being as it was a good review video for some of the translation and all of that, I'll let it keep going. But what we need are, okay. Boom. And it came right up. Okay. All right. And just Okay, I'm gonna try to get this one to share. All right, today we're just about vocabulary. All right, so the last one talked about eukaryotic cells, prokaryotic cells. You need, well, where'd my Zoom go? I said, and in the middle of that last one, it asked me if I wanted to report to Zoom. So did the Zoom stop for you all or something? It didn't stop for me. It might have stopped for you. All right, in any case. All right, so this one is definitely going to, so they talked about the gene expression, whether the gene is going to be expressed or not. So now she's going to be looking at, the Meba sisters will be looking at, what do we call it when we express it or we
Messy,
don't?
unable to decorate my classroom kind of perfectionist. Actually, maybe I'm not a perfectionist. I wanted to make my classroom inviting and exciting, and I really wanted my students to walk in and think, this is amazing. I could never make it look like what I imagined up. But the most amazing thing did happen in the week to follow. I knew a colleague who had a friend who had a son who was going to college. Confusing. But anyway, that son could not take his pet snake to college with him. Some rule about dorms and snakes. My colleague asked, would you like it? They said they can give you the cage and everything. It could be a classroom pet. I didn't even have to think about it.
Skip.
Of course, amazing popular one at that. He was friendly, well, for a snake. He'd let you hold him and not bite. That year I was the only one in the science wing with a pet snake, and I'm pretty sure there were some people that wondered, what was the point? Well, I'm big on relevance, so anytime I would teach a biology concept, I found some way to work in Spike. Predation? Well, let's talk about Spike's appetite for rats. I expect an answer. It was a great question because we were getting close to our heredity unit. Heredity is about how traits are passed down from parent to offspring. We've made a playlist of our videos that focus on heredity, including reproduction, how to track inheritance and pedigrees, how to solve genetic problems in Punnett squares, and understanding different Mendelian and non-Mendelian
We need
inheritance.
to talk about all those things. There's a lot.
But you really can't delve into those and study heredity without understanding DNA, chromosomes, genes, and traits. And that's what this introductory video is going to focus on. So back to the student question. Spike has traits. The patterns on his body, his size, these traits are coded for in his DNA. Some of the traits he inherited can be influenced by the environment. For example, if Spike had not had the nourishment he needed, that could affect his size. DNA is not just one big code buried deep down in an organism like some treasure. Spike's DNA is in the nuclei of nearly all of his body cells. He inherited his DNA from his mother and father. I can't know for sure what Spike's parents look like, but I do know that Spike inherited his DNA from them. One fun fact, many snake species can reproduce asexually. Had that been the case for Spike, he would have inherited all of his DNA from only one parent. But it would still be DNA coding for traits. Just like Spike, your DNA codes for your traits, and your cells can't function without it. DNA determines how tall you are, what color your eyes are, what color your hair is, or even if you're at risk for certain diseases. Also, like Spike, your entire DNA code is in most of your body cells. That's why in those crime-solving shows, which we may or may not absolutely love, a criminal can sometimes be caught by just leaving a cell from a hair follicle behind. DNA has a beautiful structure, and that structure will help you understand how inheritance works. DNA stands for deoxyribonucleic acid. It's a type of nucleic acid. If you remember from our biomolecules video, nucleic acids are a type of biomolecule. Nucleic acids are made up of building blocks called nucleotides. Nucleotides have 3 parts. One of them is a sugar called deoxyribose. One of them is a phosphate. We sometimes say DNA has a sugar-phosphate backbone. But the most important part of the nucleotide is the base, because the sequence of the bases can actually code for traits. So as far as the bases go, there are 4 types of bases in DNA. A lot of times they'll just use the letters A, T, C, G. The A is for adenine, the T is for thymine, the C is for cytosine, and the G is for guanine. These bases actually pair in a specific way, and there's a popular mnemonic that can help you remember which of them pair together. Apples in the tree? That tells you the A for apples, T for tree, because the bases A and T go together. The other verse is car in the garage, and that can help you remember the base C always goes with the base G. Regardless of whether we're talking about Spike, or a plant, or a protist, or a human like you, these are the DNA bases in living organisms. But the amount of DNA bases overall, and the sequence of those bases we mentioned, will vary among different species, and also among different individuals. But it's likely to infer that Spike has a sequence of DNA bases that is more similar to his parents than he would to, say, a rattlesnake. DNA has two strands, so there are nucleotides running up one side and there's nucleotides running up the other side. The bases are what pair in the middle. The bases are held together by hydrogen bonds. The DNA is also twisted in something we call a double helix shape. Portions of DNA make up genes. We can say, for example, that this part of the DNA here makes up a gene. Genes can code for proteins. Proteins have a huge role in expressing a trait. For example, let's consider your own eye color. Human eye color is actually pretty complex. It's determined by many genes. The genes can code for proteins involved in producing the eye color pigment, but proteins coded for by genes, they can play a wide variety of roles besides just your eye color. Proteins are involved in transport, in structure, in acting as enzymes that can make all kinds of materials, in protecting the body, and so much more. We do want to mention that not all genes are used to make protein, and there are parts of DNA that are non-coding. And even though nearly all of your body cells have your entire DNA code, your body cells may only use certain portions of those genes. Genes can be turned on or turned off by a variety of mechanisms. We call that gene regulation. Check out our video on that. Now, you have a lot of DNA. When it's compacted, it can be organized into a unit called a chromosome. Very helpful when you're trying to make more cells and you need to get the DNA into those new cells. Chromosomes in your body involve DNA wrapped around protein structures. Humans have 46 chromosomes. That means nearly every body cell in your body has 46 chromosomes. Human sperm and egg cells, on the other hand, each contain 23 chromosomes. So you receive 23 chromosomes from your mother, and 23 chromosomes from your father to give you your 46 chromosomes, your genetic code. So let's do a recap of the big picture. Here's a single chromosome. You see genes on this chromosome. These genes consist of portions of DNA. DNA is made up of nucleotides, and it's these bases here, the sequence of them, that makes the difference in coding for traits. Whew. Understanding this foundation is essential for understanding heredity, whether you're talking about you or Spike. So this may bring up more questions now, like where do dominant and recessive traits come in? What about alleles, and how do you arrange those around a Punnett square? Well, you can explore those concepts and more in our heredity playlist. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Speaking of the heredity playlist, that's on the playlist. So that was review up to a certain, so up to that point, you should have already known, because that took us up to mitosis and meiosis, where we talked about the genes are wrapped around a protein, and chromatids and chromosomes. So now we need the next one. Okay, the numbers that you need to remember. Hold on, give me 1 second. I do want to save this one. All right, then I want them in chronological order. which they are not. All right, today is just going to be a vocabulary kind of day. All right, this one might be the next one in succession. All right. They're not in the order that they're supposed to be in. Because normally they just go into what should be next. And I'm still trying to get them to talk about dominant and recessive, and she keeps talking all around it. All right, so this one is closer to where I want to go.
I don't remember which grade it was where I learned something about my taste buds that can never be unlearned, but the event in the lesson with genetics has stuck with me forever. For you see, I learned that my taste buds cannot taste PTC. PTC stands for this. We'll stick with PTC. And it's a chemical that can be sold on these paper strips. It can be purchased under the name PTC paper, and it's popular in genetic classes because it has this fascinating quality. Some can put it on their tongue and immediately say, yuck, this is bitter. And some people, when they place the paper on their tongue, taste absolutely nothing. Well, unless you consider the paper. Does paper have a taste in itself? That's a debatable question, but the point is some people can taste PTC and some people cannot taste PTC. And I was really disappointed because I remember that I was the only one there that could not taste it. So there was everyone getting this amazing science experience and I couldn't taste a thing. Well, there may have been more than just me that couldn't taste it in the classroom that day, but they didn't seem as concerned by the fear of missing out of the PTC paper as I was. I remember someone trying to make me feel better, saying, "Oh, but it tastes bitter." For myself, what it would have tasted like. Of course, the reason PTC paper is used in genetic classes is because the trait of being able or not being able to taste PTC is based on genetics. A reminder from our Intro to Heredity unit that genes are portions of DNA, and they have the ability to code for a characteristic, a trait, like being able to taste or not to taste PTC. Now, we do want to point out that many traits are actually coded for by interactions of more than one gene, like eye color, which is quite complex and determined by interactions of many genes together. In fact, the ability to taste PTC or not may involve some other gene interactions. There's even different ranges for how bitter the chemical may taste because there may be more kinds of alleles than we'll mention. More about that later. But since we do know that the ability to taste PTC or not taste PTC is at least heavily impacted by a specific gene, it does make it powerful for genetic classes. One thing I have found so interesting is that my parents can both taste PTC. So why can't I? Recall that humans have 46 chromosomes. Chromosomes are made up of DNA and protein. It's a condensed unit of DNA. My whole genetic code is represented by these chromosomes. You inherit 23 chromosomes from your mother and 23 chromosomes from your father. Here's all 46 of them right here. As you can see, there are 23 chromosome pairs. Each pair has one chromosome from one parent and one chromosome from my other
this
parent.
is what we're going to get into looking at. We're going to look at this is called a karyotype or a chromosome map. So we're going to be looking at these. We're going to notice how they're all a little bit different. So we're going to be looking at what's on chromosome set 1 and which on chromosome set 9, that sort of thing. When we did the mutations last time, we talked about there being a mutation on chromosome 23 or chromosome 21. And so we'll still be talking about those. And notice you have, this one is, anybody tell me just by looking at this whether this is a boy or a girl? Do you know? I know we haven't covered it yet, but just kind of, this is what this is, see what you already know. Can you tell by looking at this if this is a boy or a girl? You can, but can you? Anybody want to volunteer? Is this a boy or a girl? I know yes, but I'm asking you, is it a boy or a girl? Yes, you can tell. I'm asking you to tell me, is this a boy or a girl? It's a girl. Why? How do you know that? All right, there is no Y chromosome. And as Isaiah is answering this, and that's exactly this one right over here. There's no chromosome over here for Y. The two chromosomes are for the X. So that means there's an X and an X. Huh?
A female.
This is a female because there is no Y chromosome. All right. If this was a boy, they would have one chromosome over the X and one chromosome over the Y. All right. So, and you'll be asked that some activities that we're going to do later on, you're going to have to be looking for stuff like that. And you're going to have to map some chromosomes too. All right. Let me try to get this thing playing again. Whoops. What did I do? Okay, we were here. Up. Too far. Too far. Where was it? Oh, yeah, that's where we were. We were right there. Now, if I can get this to play.
From my other parent. If we focus on one of these pairs of chromosomes where the PTC taste sensitivity gene may be found, we can see an area where the PTC taste sensitivity gene could be. Let's assume this is the locus where the PTC taste sensitivity gene is found. See how it is pointing to a specific area here? That's because it's on an area of the chromosomes that refer to a specific gene that codes for a trait. Now, remember how this chromosome is from mom. This one is from dad. Each parent contributes an allele, which is a variant of a gene. An allele is a variety of a gene, a form of a gene. The alleles could be the same form of the gene or different forms of the gene, but regardless, in this case, they're forms of the gene involved with PTC taste sensitivity. So, if PTC taste sensitivity is being used as a one gene trait example, and as we mentioned, it may not be that simple, then your DNA code has a gene related to PTC taste sensitivity. Together, the two alleles you inherit, the forms of that gene, determine the trait of tasting PTC or the trait of not tasting PTC. That gene is involved with coding for taste receptors on your tongue, and the receptors you have can make a difference for whether you taste PTC or not. The alleles are typically represented by letters. Since this is all about tasting, let's use the letter T. But wait, it matters whether I represent it as a capital or lowercase letter. If I use a capital letter to represent an allele, it means that it's a dominant
Right, these
allele.
are the rules for expression. So you're going to remember that. Capital letters are for dominant, recessive letters are, lowercase letters are for recessive. That's a rule. It's going to be important. And notice what she did too. She picked the letter that represented the trait. So she's looking at taste. So they use the letter T. That's also going to be important because when you all have to do this, you're going to have to pick the letter that goes with the trait. You're going to have to decide whether it's dominant or recessive, whether you're going to use a capital letter or a lowercase letter.
If one or both of the alleles you inherited for a trait are dominant, then it will be expressed. More about that later. If I use a lowercase letter to represent an allele, that means it's a recessive allele. Recessive alleles are typically not expressed unless there is no dominant allele present. Now remember that you have two allele copies, so the combination you could have here could be big T, big T, or little t, little t. These are called genotypes, your genetic makeup. Genotypes can help determine a phenotype, which is a physical characteristic. You'll notice when writing genotypes, I put the capital letters first, if it contains a capital letter. That's not because the order matters, it's a formatting formality that capitals are written first. It turns out that being able to taste PTC is a dominant trait. That means the phenotype, which is a PTC taster, is due to a genotype that includes at least one dominant
We're
allele.
supposed to
So
show
which
first.
genotypes can taste PTC
But
then?
the other ones were good for review. All right, so we're going to start with this next class, and then I'm going to have a worksheet for you to work on practicing recognizing dominant and recessive traits, and using capital letters and lowercase letters to represent those things. So that's what we're going to work on next class. We're going to look at the terms phenotype, genotype. We're going to look at alleles, all of that next class. Okay, so vocabulary. If you do nothing else between now and then, look at your vocabulary, because it's going to be important that you understand what the word means. Because when I ask you, what is the phenotype, you need to know what I mean by that. When I ask you, what is the genotype, I need you to know what I mean by that, because that's the way the questions are going to go. All right. Okay. You all have a great rest of your day, and see you on Wednesday. Wednesday is, we're having a short week, so Wednesday is a regular day. Okay. Bye.